Bonjour, je voudrais commencer par remercier France Parkinson pour m'inviter, pour présenter mes recherches et aussi pour
Hello, I'd like to
vraiment
thank
organiser
Franz
un événement
Parkinson
de
for
ce
having
type
organized this
si
research.
important et puis pour participer au
It's very
financement des
important
recherches euh, pour nous et pour nos étudiants.
for us and
Alors moi,
for
je vais
our
vous parler
students.
du projet qui est justement financé par
I would
France
like to
Parkinson,
discuss
qui
the
est
project
comment
being funded
comprendre,
by Franz
comment
Parkinson. How do we
on essaye de
understand
comprendre comment Parkin protège nos
how
cellules du stress lors de
Parkin
l'exposition
protects
à
our
l'organoïde
cells from
avec
stress
le modèle organoïde.
with the organoid model?
Understanding how
Et
Parkin
j'appuie
protects
sur
our
le
cells from
bouton maintenant
stress
et c'est
and
bon.
mitochondria using the
Et
organoid
donc,
model.
nous, on essaye de comprendre, comme beaucoup d'autres,
We're trying to
pourquoi les
understand
neurones dopaminergiques de la
how,
substance noire dégénèrent majoritairement dans la maladie de Parkinson.
how this degrades
Donc,
into
vous le savez,
predominantly in
lorsqu'ils
Parkinson's
dégénèrent, eh bien,
disease.
ça va
Why do dopaminergic
introduire l'apparition
neurons
de
degenerate?
dysfonctionnements
The
moteurs
appearance of motor
assez importants.
dysfunctions
Il
of
y a
significant
plusieurs
proportions. There are
hypothèses
several
qui sont
hypotheses
amenées par
that have been put
pas mal
forward
de scientifiques dans le
by many
monde. Une
scientists in
des
the world.
hypothèses est
One of
que
those
les mitochondries, qui sont
is
les
that the
usines de
mitochondria,
fabrication d'énergie des cellules,
which are energy producing
dysfonctionnent
factories
dans
of
la
cells,
maladie de
are not
Parkinson
working
et cela entraîne
properly.
la mort
There's
des neurones
a
dopaminergiques.
defect in energy production.
Il y a un certain nombre
There
d'indices
are a
qui
certain number of
permettent de penser
indices
cela.
which
Par exemple,
lead to that
eh
idea.
bien,
For
dans des
example,
échantillons
in
humains, on s'est rendu
samples,
compte qu'il
in
y a
human
des petits
samples, we
éléments
realized that there
de la
are
chaîne de fabrication d'énergie de la
parts of
mitochondrie
the
qui
energy
fonctionnent
producing
pas très
factory,
bien.
as it were, of
Et
mitochondria
puis,
that are not
il
working
y a des
right.
agents toxiques dont vous
And
avez
there
peut-être
can be
entendu parler,
toxic
comme la
agents, you
réténone,
may,
roténone ou MPP+,
such as retinol
qui
or
ciblent
MPP+,
spécifiquement justement ces
which
petits
target
éléments de la chaîne de
um,
fabrication d'énergie.
parts of the energy
Et donc ces
production
éléments, eh bien,
activity in mitochondria.
ces agents provoquent
And these
l'apparition
agents
de
can bring
symptômes
about
comme ceux qu'on
symptoms
voit
that
dans la
are
maladie de
reminiscent
Parkinson.
of
Enfin, il y
Parkinson's
a des formes
disease.
de la
Lastly,
pathologie qui sont des formes
there are
génétiques
forms,
familiales
genetic
associées
forms
à
of
des mutations
familial
dans des
type
gènes
of
comme
mitochondrial proteins
PINK1
involved,
ou Parkin.
such as PINK1 or Parkin.
Or, ces gènes codent des
These
protéines qui
genes
sont dans la
code
mitochondrie
proteins that
et
are in
qui
mitochondria
sont
that are
responsables
involved in
un peu du
mitochondrial
contrôle qualité de
quality
ces usines
control,
énergétiques des cellules.
including
C'est-à-dire qu'elles sont
mitophagy. In other
à
words,
la
in
surface des mitochondries, elles sont
familial
capables de
form
sentir
of
quand
Parkinson's. So
les
they can actually
mitochondries se
feel
sentent pas très
when the
bien,
mitochondria are not
premièrement,
feeling
et
well.
puis elles vont signaler à la
And they
cellule,
will
OK,
be
là
indicating to
maintenant,
the
il est
cell,
temps
okay, now it
de
is time
dégrader la partie, enfin
to
oui, de dégrader la partie qui est endommagée
degrade
pour s'en
the
débarrasser et
damaged
garder la
portion to get
partie
rid
saine,
of it and
ce
to
qu'on
keep the
appelle la
healthy part.
mitophagie.
And we call that mitophagy.
Donc, nous, comme beaucoup d'autres, pensons que
We
si
believe
les
that
neurones dopaminergiques
if
dégénèrent dans la maladie de Parkinson, c'est
dopaminergic
parce qu'ils ont une
neurons intervene, it's
vulnérabilité
because they
accrue
have
à un
an
stress
increased
dû au
vulnerability to
dysfonctionnement
stress. There's a bit of a
des
dysfunction
mitochondries.
due to the
Mais il y a
mitochondria.
deux interrogations quand
But
on
there
dit
are
ça.
two
La
questions.
première, c'est que je vous ai
The
parlé
first
des acteurs moléculaires, donc
is
des
that the
petites
molecular
protéines
players identified to
PINK1
date,
ou Parkin,
PINK1 or
mais
Parkin,
ce sont des
are
protéines qu'on trouve dans toutes les
present
cellules,
in
pas que
all
dans les neurones
cells, not
dopaminergiques.
only
D'accord?
in dopaminergic
Pourtant, dans la maladie
neurons.
de Parkinson, ce
However, in
sont
Parkinson's,
principalement les
it
neurones
is mainly
dopaminergiques qui dégénèrent.
the
Et même
dopaminergic
la
neurons that
réténone ou MPP vont
generate.
viser les mitochondries de
The,
n'importe quelle cellule. Or, les neurones
but there are molecular
dopaminergiques
mechanisms specific
vont
to
dégénérer
these that cause
principalement
these
là.
neurons to be
Donc,
particularly
nous pensons
vulnerable to mitochondrial
qu'il
stress.
existe des
So, what are the
mécanismes
mechanisms
moléculaires propres aux neurones dopaminergiques qui font qu'ils
proper
sont
to these
encore plus
dopaminergic neurons,
vulnérables à des
which makes them
stress, des
more
dysfonctionnements
vulnerable to
mitochondriaux.
stress?
Mais on ne sait pas
Or, but we
ce
don't
qui fait cette particularité. On ne
know
connaît pas ces
why it's
mécanismes.
different. We don't know those
De
mechanisms.
même, ces
In the same way,
protéines, donc PINK et Parkin,
Pink and
elles sont
Parkin
exprimées de façon précoce dans la vie.
are
Ce n'est pas quelque chose
stress
qui arrive
sensors. They're
avec
precocious.
l'âge. Et donc, on peut se demander, est-ce que
We can
lorsque
wonder
celles-ci dysfonctionnent,
if these are
comme quand on a une mutation,
dysfunctioning,
est-ce
as in
que
the case of
ça
mutation.
entraîne également
Does
des
this
défauts
automatically
biologiques, des
bring
petits
about
défauts dans les cellules
degeneration of
euh,
DA neurons in
tôt
Parkinson's
dans la vie,
disease
mais qui
early
sont
on in life?
silencieux, dont on ne se rend pas
But
compte
they're
tout de suite.
silent because you don't realize that they're there right
Alors,
away.
nous essayons, donc quand je dis nous,
We are trying.
c'est Christiane Zuck, qui est une doctorante
Christian
du
Duke, who's
laboratoire,
a doctoral candidate in the laboratory. If you'd like to meet her, there's a poster of her here. We're trying to understand how Parkin plays a role in protecting cells faced with this type of dysfunction of mitochondrial stress, and if the role is different depending on the cell type, what means that the neurons are more or less sensitive to this. We believe that Parkin and Pink act as a mitochondrial stress sensors, and they can coordinate various protective responses in cells, not only mitochondria, but a whole set of responses to protect the cell from these stress. So Christian's objective is to identify the molecular mechanism triggered in dopaminergic neurons following mitochondrial stress, and to evaluate the dysfunction of these stress responses and see if they're different in other cell types that we have in the brain, and obviously to assess the dysfunction in the context of the Parkinson's disease, for example, in the absence of Parkin. So to do this, we use a study model. It's quite recent, which we call mini brains. They're more organoids that we obtain thanks to stem cells that are pluripotent, and they are induced pluripotent stem cells during development. In the uterus, we have stem cells that are embryonic, that have the power to become any type of cell. They can become, as you can see, over time, they will be differentiating into different types, and little by little, these cells will be specialized to become skin, pancreas, lung, and even in the brain, there are many different types of cells. So this is differentiation. Now, and it's only recently that we are able to go backwards. In other words, starting from these specialized cells, we can reprogram to back up and become some cells that are pluripotent. In other words, they have the power to re-become many other things. And yes, it's induced because we program them that way. So in practice, how does it work? Well, we take a skin or a blood sample from the patient. And based on that, we reprogram to have stem cells that are pluripotent from the patient. And these stem cells, we can then if in the presence of a certain milieu, you can obtain dopaminergic neurons or skin cells or heart cells or whatever. We try to obtain cerebral tissue, and we try to obtain something that is close to the region that gives notion to black matter, the ventral mesocephalus. What you see on the left, like the size of lentils, these are the organoids. And we are able to cultivate these cells so that they get self-organized into 3 dimensions, and they're adopting an organization similar to that which we observe in different regions of the brain. There's an example in the middle. This is a cross-section of an organoid. You see a superficial layer, and you see at the beginning of the development in the cortex. And the advantage of this method is that we will obtain neural networks that are much more mature than when you do 2-dimensional cultures. This approach, using these organoids, makes it possible to do many things. This is a modulatable technology because using these induced pluripotent stem cells, we can generate organoids from different regions of interest. We can have organoids from the central ventral midbrain organoids, where the substantia nigra originates, or subcortical organoids from the striatum, etc. And with blood samples or EPS, we can obtain brain immune cells. And with all of these, we can assemble these different regions. and we call these assembloids. The idea being to see, for example, if these regions can be connected as they do connect in real life. In this context, Christiane, who is studying the response of dopaminergic being exposed to stress, she uses took induced pluripotent cells, which were not expressing Parkin, and she cultivated them so that they become organoids, either in basal condition or in chronic stress condition. In other words, a bit of a regular stress where we are going to help the manufacturing chain to malfunction a little bit, but constantly. And after 60 days, in other words, 2 months of culture, she has done a lot of experiments about fluorescent labeling and various highly developed techniques for each cell to see what genes are expressed in these cells as a response to stress. And her objective was to compare the mitochondrial stress responses in dopaminergic neurons versus other cell subtypes, and to see if the alterations in these responses in the context of Parkin deficiency. So I'm going to show you these preliminary results. They're from last week. Looking at all of this, on the left, you see images of cross-sections of organoids. And so we're able to mark the cell types. We can mark on the red at the top, you have neuron progenitors, dopaminergic neuron progenitors. In the middle, you have GABAergic neurons, and at the bottom, you have astrocytes. These are other brain-type cells. In reality, what we observe is that the controls and organoids that don't have Parkins have different cellular composition of ventral membrane organoids in the absence of Parkin, much less marking. And then she's continuing her analysis because it's a lot of work. Single cell sequencing to see the expression in each cell, the expression of each gene. So this is represented by this diagram you see here. Each point is a cell. And by looking at everything that's expressed in those cells, we can actually say, give the identity to the cells to say such and such a subtype we have. So they're represented by colors that reflect that. The red ones, it's PDA. These are progenitors, dopaminergic progenitors. And so what do you see? You have in the organoids that do not have Parkin at the bottom, there are more dopaminergic progenitors in the controls. Similarly, we have differences in level of the subtypes, where there are fewer, which demonstrates that we have an alteration of the cellular composition of the organoids in the midbrain organoids in the absence of Parkin. In other words, Parkin could play a role in the very upstream developmental processes. Confirmed, obviously, with different additional experiments in different types of cultures. These differences may be compensated over time. So finish with the results. What I showed you was a basal condition. The stress response of mitochondria, we're seeing that chronic stress increases cellular alterations in the absence of Parkins. So the organoids that have no Parkin will have more dopaminergic progenitors than the others, and less dopaminergic. But when you look at the whole set of the responses, we see that there's a difference in the treatment. In the controls and in the ones that have no Parkin, stress brings about a more important effect on the mitochondria. You can see that presented by the green arrows. And in controls, there's also going to be a protection effect of proteins. In other words, if by stress the proteins are not well folded or whatever, we have something that refolds them properly. So in the organoids without Parkin, the process is not there, but it's even deactivated. So one response to stress, which is dysfunctional. Now, what remains to be done is to compare these responses to stress with responses in other cell types. I'm going to conclude by explaining that this organoid approach to studying the molecular mechanisms involved in Parkinson's disease is using human cells. So there's possibility of working with patient cells. It can also be used as a test platform. We can assess different types of molecules for different therapeutic approaches and confirm the genes identified by genetics. I showed you the example of Christian, who's working on a stress response, but we also have Elena in the laboratory who's been working on neuroinflammation in Parkinson's disease, Elena Winterberg. You can see on the video. We also have Pauline Cheval, who has a poster here if you'd like to see. She's working on the establishment and functioning of neural networks, the assembloids, in presence or in absence of Parkinson's. And we can also work, as Annelle is working, to work chimeric organoids to validate the genes associated with Parkinson's disease. I hope I stayed in the time. In any case, thank you so much for your attention. I'd like to thank my team. It's a kind of a big team. There are a lot of us. And the organoid pole. And you have quite a few members here today. Thank you.
Merci beaucoup. Vous m'entendez toujours? Oui, merci beaucoup. Donc, vous comprenez, là, on est vraiment dans de la recherche très fondamentale, dans les mécanismes.
This is really fundamental research. It's not always easy to explain, but I think it's important to realize that this research does exist. It's necessary so that we can do some research on clinical molecules. So thank you. It was very important to talk about these questions, even though it's a bit more complicated to explain. We have a few questions that we've prepared. Perhaps we could have the first question, please. We talked about organoids. Why do researchers use organoids in studying Parkinson's disease? To model small human brains to replace MRI or to look so that it looks pretty in the labs. Okay, we're trying to relax you guys. A little transition here.
Je pense qu'il y a des gens qui nous font quand même des clins d'œil comme nous on le fait. Donc voilà, donc n'hésitez pas, après il y aura tout le temps de poser des questions aussi sur ces
You can always ask questions on the organoids.
Ce ne sont pas des vrais mini-cerveaux comme vous l'avez dit.
They're not mini brains, as you may have realized, but we could talk about that afterwards. Thank you.